Okay. Now we talked about these guys, being made. How do you break them? Well, CMP is converted essentially to, let's say, beta alanine or alanine, and this one is also converted to, at the end of the day, they're both converted to acetyl CoA. I'll just keep it simple. Okay, acetyl CoA. When we reach to that level, we'll talk about it. So they don't produce anything toxic. They can be used directly for, they can be used for ATP synthesis. Okay, so they're used in that sense. So really there's not much to it in this case. Especially, for example, U and C are converted to alanine, and this one is converted to aminobutyrate. So it isn't, at the end they're both converted to acetyl CoA. And acetyl CoA, if you need energy, will be consumed for energy. You can further make it to convert it to oxaloacetate and make sugar, or store it as lipids. So it depends what we have. We'll reach to that point. Essentially, once we study glucose and some other amino acids and fatty acids, you will see that there's a central molecule combining all of them, merging everything together. So that means there's interchange. Yeah, there's interchange. So if you have too much amino acid, they'll be stored as lipids. If you have too much amino acid and you have lack of energy, they'll be used as energy. So it depends. Okay. All right. So there's nothing specific in this case, but just please remember this: B12 affects this area, folate affects this area. Okay. And again here, tetrahydrofolate. So we have to think about those things, and we should be okay. Now, when you're thinking about this, if you want to know further evidence, or just you feel like you're not getting it, number one, they are deaminated. Okay, so they're deaminated. So there is an amino group removed, and then the sugar is removed, and then the ring is opened, and then further deaminated. So there's multiple steps involved, but we don't care for them. Okay, if you start memorizing each step, we'll never end up becoming a doctor, right? So you're not becoming a biochemist. So just the basic things that are related to diseases, that's where our goal is. If you have extra time, master what I told you first. If you have extra time, then please knock yourself out with information and stuff. It's a good idea.
So it's deaminated, then it's R5B again.
Then sugar is removed, and then the ring is opened up. When the ring is opened up, will become, the amino group is removed further, and the carbon dioxide, the carboxyl group in carbon are used as acetyl CoA.
And it's the same for both sides?
Yeah.
What's the open ring called?
Beta alanine. The end result is beta alanine. And then beta alanine is converted to alanine, and then alanine will be used to convert to pyruvate. Pyruvate can be used to make glucose or acetyl CoA. It depends what you want to do. If you have hypoglycemia, again, I don't want to go too much into detail because it will come. We have to know sugar metabolism first. We have to know TCA cycle, Krebs cycle, and we have to know lipid synthesis and so forth. But at the end, this is the central molecule for everything. At least memorize this: acetyl CoA. Okay. There's some amino acids, so there's about 20 amino acids total, right? 11 of them are non-essential, right? And then about 9 of them are essential, right? Amongst those 11, that means all 11 of them can be made from your body, by your body, and they can be converted to glucose or ketones. So there's some amino acids that are ketogenic. Ketogenic amino acids mean they're converted directly to what? Acetyl CoA. Glucogenic, they're converted to what? Glucose. Okay, we'll talk about them too. So the idea of this cycle was for you to learn that number one, nucleotide, how they're made. How do you make nucleotides? That's one thing. And the second thing is, how do you break nucleotides? Well, at least one of them is xanthine and uric acid. This is important. The other one I wouldn't care for. Okay. But in nucleotide synthesis, there's amino acid involved, and tetrahydrofolate and B12 is involved. So nucleotide synthesis depends on folate, on B12, and on amino acids. Okay? Alright, yeah. Hani, you had a question?
Yeah.
Hypoxia. Yeah, let's do that. Good job. Good job, good job, good job. So hypoxia. What does hypoxia mean?
Decrease oxygen in tissue.
That's 100% correct. What is hypoxemia? Blood. So there's different. See, hypoxia means the tissue is not getting oxygen. Does not mean it's low in blood. Okay, so when you say low oxygen, what does it mean? Is it in air? Is it in my body? Is it tissue? Is it blood? Is it RBC? Is it heart? Is it lungs? Hypoxia means tissue is not getting enough oxygen. Tissue. Tissue, tissue, tissue. At the end of it, that's what matters. Right? So hypoxia means tissue is not getting enough oxygen. But you can have hypoxemia, which is lack of oxygen where? In blood. Okay? Yeah. You can also have the result of these two, and that is cyanosis. What does that mean? Blue discoloration of the skin because of lack of oxygen. Very good. Okay, so hypoxia. Let's say hypoxia means low oxygen. Now, why is low oxygen a big deal? Look, the most important thing for life is oxygen, right? We've seen this many times. You can live without food for days. You can live without sugar for years. You can live without amino acids for years because something else will compensate. Without lipids, without vitamins, without this, because people are vegetarian, vegan, and blah blah blah. We have all the examples in life. They're still living. Maybe they're not happy, but they're living nonetheless, right? Now, there is no human being that I know of who can live without oxygen for more than 5 minutes. You can't. So it is essential for living. Therefore, one of the major causes of cell death or injury is what? Hypoxia. Major cause of cell death is hypoxia. And we talk about necrosis already. Okay? Alright, so what did I say? In what context I'll talk about hypoxia? Okay, let's see what I can do. So if hypoxia, that means the basic things that what happens with hypoxia. Oh, I remember erythropoietin. Let me write it down here. Erythropoietin. So I'll remember that to add to this. Now, when there is hypoxia, of course, the organelle that uses oxygen is mitochondria. Mitochondria. So we have two processes that produces energy. One is glycolysis. Okay. Glycolysis, which means breakdown of glucose, and the other one is oxidative phosphorylation. Oxidative phosphorylation. Okay, oxidative phosphorylation. Now, oxidative phosphorylation occurs in the mitochondrial matrix. Of course, it has, some enzymes that are on the membrane as well, but primarily on the matrix, or especially on the inner membrane. The electron transport chain is located on the inner membrane. So oxidative phosphorylation means electron transport chain. Electron transport chain. Electron transport chain. Okay. Whereas glycolysis occurs in the cytoplasm. Okay, and then we see how hypoxia affects both processes.
Where does the Krebs cycle play?
Matrix of mitochondria.
So is that part of the oxidative phosphorylation?
Okay. You know what? Once I finish this, I'll start with that. Well, let's start with it because I think that's the core of everything. Because sometimes I assume students know it. I'll go over that. It will make life much easier for other processes for you to understand them. Okay, so look, oxidative phosphorylation occurs where? In the mitochondria. In the mitochondria. Okay. Just keep it simple. So in the mitochondria, even matrix is, you can remove it. Now, glycolysis in the cytoplasm. In cytoplasm, the environment is anaerobic. The environment of cytoplasm is anaerobic, meaning there is no oxygen. There is no oxygen available in the cytoplasm. In the mitochondria, the environment is aerobic, meaning there is oxygen available. So whenever we breathe oxygen, it is simply to replenish the concentration of oxygen in mitochondria. Okay? Alright. So at the end of the day, if we need energy. We need energy. Each cell, for it to survive, it needs energy. What is the definition of life? That's the purpose, not definition. That's the purpose, maybe. Okay.
Be able to use energy.
Living things. How do you define something that's living versus non-living? How do you differentiate between this phone and me? Metabolism, regeneration, or generation of energy. This cannot generate energy. You have to plug it in. I can generate energy by myself. So living things can generate energy. They can they can have chemical reaction occurring in metabolism, and they can reproduce. I never seen the cell phone having children. I do, right? So reproduction is a big deal when it comes to life. Use of energy and reproduction of energy. So now, in order for cells to live, they better produce enough energy to survive and function. They should have a mechanism for cells to live on their own independently. You see, living things must have independence. Of course they have, depends on certain, but overall, okay, for them to function independently, they must have a mechanism to produce their own energy so they can run their own processes. So with the help of aerobic respiration or oxidative phosphorylation in the mitochondria, overall from a single molecule of glucose, since glucose is the major source of ATP or the primary source, okay, from one molecule of glucose you produce, roughly speaking, I'll go to the maximum, 38 ATPs. Okay? You were here early, right? Yeah. Okay. Yeah, just bugging her. From a single molecule of glucose, without oxygen, you can only make 4 ATPs, but guess what? Out of 4, 2 are used. So let's just make it 2 then. Total ATPs, net ATPs produces 2. If you divide 2 by 40, what percentage is that? 4%. Right?
5%.
5%, sure. Can you survive? Can your phone survive with 5% of charge? Especially if you want to watch a video.
Not too long.
Not too long. So let's give it maximum to 5 minutes. Let's just say 5 minutes. 5% of energy gives you 5 minutes of life. 38 ATPs gives you a lot more longer life. So that's the major difference, okay? Now, in the case of hypoxia, when we have hypoxia, so how would hypoxia affect oxidative phosphorylation and how would it affect glycolysis? Now you know this, the purpose of each. Let's further move on to oxidative phosphorylation. So, which means oxidative phosphorylation will decrease without oxygen, correct? Yeah. And glycolysis is a compensatory mechanism. It should increase because now instead of everything moving towards oxidative phosphorylation, all the glucose will be consumed as what? Glycolysis. Okay, so I should say anaerobic glycolysis. Especially, let's put here anaerobic glycolysis. Anaerobic glycolysis. The enzyme that primarily drives oxidative phosphorylation is known as pyruvate dehydrogenase. The initial one. Okay. The enzyme that drives glycolysis is known as lactate dehydrogenase. Pyruvate dehydrogenase depends on oxygen. Lactate dehydrogenase depends on lack of oxygen. L for lactate, L for lack of oxygen. So if you have oxygen, everything will shunt towards what? Pyruvate dehydrogenase. If you have no oxygen, then glucose and sugar will be metabolized with the help of lactate dehydrogenase. So lactate dehydrogenase should increase. Its activity should increase when? When there's hypoxia. Very good. Now, lactate dehydrogenase, the product of lactate dehydrogenase is lactic acid or lactate. Okay, if you don't like this term, I'll make it lactic acid. Same thing. Lactic acid increases as a byproduct of this pathway. And when lactic acid increases, this leads to increase in what? Or decrease in what? pH. Decrease in pH. Now there's acidity and there's not enough ATP in these conditions, so the cell, things are very, very sensitive to low pH. Are enzymes, proteins, protein, enzyme, same thing, right? And then of course DNA and RNA. Are ribosomes protein? Yeah, it's a combination of protein and RNA. So lack of pH causes denaturation of these guys, and this is usually in the cell. You will see chromatin condensation, chromatin damage, okay, chromatin accumulation. Okay, so at the end of the day, now with lack of with too low pH, your enzymes are damaged, your proteins are damaged, your DNA is damaged, your RNA is damaged. That's not a good thing. Okay, so that's what hypoxia can do. Just because you have increased glycolysis, and the product of glycolysis is what? ATP and lactic acid. ATP is not enough to make you survive, but at least it gives you some time, just enough time maybe to look for source of oxygen maybe. Okay, so it's a defense mechanism, an alternative pathway for you to survive, maybe one extra mechanism to survive. Okay, now in this case, so the function of pyruvate dehydrogenase decrease. At the end of the day, in this pathway you have less ATP. The product is less ATP. If the product of phosphorylation, oxidative phosphorylation defect is what, or hypoxia is what? Less ATP. But you know that there are major things that depends on ATP. One of them is sodium, potassium, pump. Okay? Sodium, potassium, pump. If you have no ATP, what happens to this pump's activity? Decreases, of course. Because this pump needs ATP. Without it, it can't function. It cannot function. I'm going to go on the side over here. This pump is supposed to, so let's say this is the pump. This pump is supposed to do what? What's the function of the sodium potassium pump? It's supposed to pump sodium out and pump potassium in. So exchange 2 potassium for 3 sodium. Excellent, right? Now, if this pump doesn't work, what happens to sodium concentration inside the cell? Increases. So let me reverse this. So as long as you understand that. Whoa. Now sodium concentration inside the cell will increase. What happens when there's too much sodium inside the cell? It depolarizes. It depolarizes. That means more sodium channels will open up. Voltage gated sodium channels will open up. True? And they stay open. They don't close because they depend on what? Voltage. They're ligand gated sodium channel, there's voltage gated. So this allows even more sodium to come in, correct? More sodium comes in, and what follows it? Chloride, which is negative, to match the positive. When these two are inside, water has to come in too. When water comes in, what happens to the cell? It swells. When the cell swells, that means further depolarization of endoplasmic reticulum's membrane allows calcium to come out. And calcium, so that's one thing, that was sodium. Lack of ATP leads to sodium pump, decreased activity, and this can cause increased calcium accumulation in the cytoplasm. Calcium causes enzyme activity increases enzyme activity, especially lysosomal enzymes. And then of course when the cell swells, the lysosomes can also swell. Mitochondria swells, endoplasmic reticulum swells. At the end of the day, this leads to membrane instability or membrane damage. When the membrane is damaged and it's porous, that means sometimes these cells can swell so much that they can get permanently damaged. If the cell bursts, it's dead. If it doesn't burst, that means lysosomal enzymes. So let's say lysosomal membrane is damaged, right? If it's lysosomal membrane that gets damaged, what happens? They produce enzymes. They're full of digestive enzymes, right? Lysosomes. And they'll start digesting what? The mitochondrial membrane, the lipases from there, the cell membrane, protease, amylase, blah blah blah. And now there is irreversible damage. At the end of the day, if mitochondrial membrane is damaged, it can release something known as cytochrome oxidase. And the cytochrome oxidase is a signal for apoptosis. Okay? If it reaches to that level, but usually necrosis would occur first before apoptosis.
The calcium being increased,
Mm-hmm.
you said?
Yeah,
And then what
in the
is it?
cytoplasm.
The enzymes increase?
Yeah, they will activate enzymes.
Like lipase and all that stuff,
Mhm.
and they'll destroy the membrane.
Yeah. Lysosome leads to necrosis. Lysosomal enzyme or damage leads to necrosis. Mitochondrial damage leads to apoptosis. Depends which one occurs first. Most of them it's lysosomal first. Okay. And the cell dies. But let's say there was lack of blood supply. Okay, let's say there was lack of blood supply for a few minutes, and then there was suddenly oxygen available. Again, now the cell can repair itself as long as lysosomal membrane and mitochondrial and cell membrane, three membranes, cell membrane, mitochondrial membrane, and lysosomal membrane. If they're not permanently damaged, then the cell can reverse. So if oxygen is available, everything will reverse. Now it has enough oxygen to make ATP. So glycolysis will decrease. Okay, so everything will go towards this way. Not a problem. But the problem is, in this case, oxygen metabolism leads to free radical formation. Now remember, if there's blood supply, there's reperfusion, right? There was cut off of blood supply for whatever reason. Hypoxia, the most common cause of hypoxia is what? Ischemia. Okay? So if it is ischemic attack, if it's not ischemic attack, no problem. But if it's ischemic attack, that means there's lack of oxygen and blood supply to that tissue, and suddenly that clot or whatever dislodges and opens up, and you have reperfusion to the same area. It can bring a lot of free radicals, and that's known as reperfusion injury. That's even worse than before, because your tissue is already weak and damaged, and you let's say there's a lot of deer, maybe lamb and stuff like that, and because of oxygen and everything, they're all knocked out and they're weak. And then at the same time, you do reperfusion, but the reperfusion brings them wolves and tigers. They're done. So reperfusion injury is like that, because the tissue is already weak, and then it's already injured, and then you on top of that, you release free radicals on them. It will cause even further damage. But I think what I wanted to say is, let's talk about hypoxia is in the cases of what are the compensation for hypoxia. Okay, so this was hypoxic damage. Hypoxia will cause damage to tissue, because you know that all tissue depends on oxygen. The reason being is what? You need oxygen to make energy. But when we have hypoxia, how would the body compensate for that? What would the body do? Is it going to just sit quiet and relax? No, it's going to do something about it. As I said, the only problem is us. When there's an issue with us, we think a lot. But the body doesn't do that. Internal environment is designed that it doesn't think. It takes action to resolve it, no matter what it is. It's tough decision making, you have to do it. Okay, so the body sometimes has to cause damage to some cells to make sure the entire body survives. Okay. Now the body will do what in those circumstances? If there's hypoxia, well, one thing you can do is, of course, whatever is involved in oxygen transport, Okay, in oxygen transport from air to tissue. One is lungs. In this case, let's say lungs output airways. Okay, airways are involved. Alveoli. Do you know what's alveoli? The air sacs in lungs, right? Airways are just those tubes, bronchi, bronchioles, and trachea, correct? Okay, so they are responsible for transporting oxygen from where? From air into blood. You also have RBCs involved in this transport. Within RBC, I'll put hemoglobin, which is used for transport of oxygen. Now further, who takes blood to tissue? Heart. So heart is involved too. Heart is the one that pumps blood. Lungs gives it oxygen. RBC carries that oxygen with the help of hemoglobin. But of course there's blood vessels too, or capillaries. So there's many factors involved in transport or delivery of oxygen to tissue. If oxygen is such a big deal, then we gotta know all of them to understand what is the result of hypoxia. Okay, if there is hypoxia, low oxygen, what would you, let's start with airways. What would airways do? Are they gonna just relax? Imagine when you need oxygen, the airways senses that. They will dilate, so there will be bronchodilation. The compensation will be bronchodilation. But further, there's, see, I keep forgetting. Let's include one more thing. Diaphragm is involved, or lungs. Diaphragm. The brain senses low oxygen in the carotid bodies. Okay? And then diaphragm contraction will increase. You'll start breathing faster. So breathing rate or respiratory rate will increase. It just simply means like you're going to breathe more and more air. So your body thinks there's low oxygen. One way you can increase oxygen delivery to tissue is to at least increase oxygen delivery from air into your body. So you'll breathe faster. And if you want to deliver oxygen from your mouth, from outside atmosphere into your lungs, you better have your airways dilate, expand. If they're narrow, less passage of air. If they're wide open, more passage. So bronchodilation will increase flow of oxygen into your alveoli. Beating of the diaphragm faster, breathing faster, you bring it much frequently, more frequently, and you exhale in carbon dioxide more frequently. So that's two compensatory mechanisms, which are fastest one. And this is by the nervous system. Okay, so CNS will signal that. CNS will signal your diaphragm and your airways to dilate, and contraction diaphragm, respiratory rate will increase. Okay. By the way, I'm just going on a side. Where is the cardiorespiratory center? In the medulla. Medulla, which medulla? Of the adrenal? Okay. Of the brain stem or brain. Very good. Very good. It's extremely sensitive. Medulla oblongata is extremely sensitive to carbon dioxide tension in blood. Extremely sensitive. Okay. So number one, you have hypoxia, medulla will get stimulated, and they will cause increased bronchodilation and increased respiratory rate. That's compensation for what? For hypoxia. Okay. Next thing. Okay. Heart. The heart now has to look. You can bring all this oxygen you want into your alveoli. If you cannot carry it from alveoli to tissue, what's the big deal? Same thing. Again, it's a problem. So the heart has to do what?
Increase.
Pump faster and harder. Okay, so heart rate should increase. So in this case, heart rate will increase. Heart contraction will increase. Yeah, all those things. Stroke volumes, cardiac output, right? And diastolic volume, everything should increase. Okay. Now, so that's in the case of heart. What is the role of alveoli? See, alveoli is the medium between oxygen and RBCs. Okay. It is the exchange area. Very good. If this is alveoli and this is a blood vessel, oxygen has to diffuse from here to here. That means blood flow to alveoli should increase. Blood flow to alveoli should increase. Okay. Number one, alveoli will dilate more. They will open up more. Okay, when they open up, blood flow to them should increase and exchange should increase. So alveolar exchange should increase. That's another mechanism. Alveolar exchange would increase. And you don't have to guess that. Alveolar exchange always means one thing and only one thing. Exchange of oxygen for carbon dioxide. Exchange of oxygen for carbon dioxide. That's what alveolar exchange means. There's nothing else. They don't exchange sugar for lipids or whatever. Only oxygen for carbon dioxide. Okay. Now, if oxygen comes from alveoli, so let's say here is the alveolus, and this alveoli is wrapped around by blood vessel, a lot of them. So oxygen in green goes from alveoli into blood. And blood, what are you going to do? There's still more mechanism. So now, wouldn't it be a good idea if you cause vasodilation? That means blood flow will be easier and increased. So vasodilation could occur. Huh?
And you can also carry more.
Yeah. So vasodilation should occur. That means more blood will reach the lungs. It's almost like having 200 taxis coming to a terminal to pick up more people. If you make it 400, you have to broaden the highways. So you have to cause vasodilation so more RBCs can come. More RBC can come. But it's still not enough. See, oxygen is carried in blood by who? By RBCs, and within RBC you have hemoglobin. That means now, in order to increase efficiency of oxygen delivery, you have to increase what? Hemoglobin synthesis and RBCs. And for RBC synthesis, you need kidney. Kidney is the one that produces erythropoietin. Erythropoietin, so kidneys are also sensitive to oxygen concentration. Whenever hypoxia occurs, kidney gets activated and they will release what? Erythropoietin. To increase what? RBCs, RBC synthesis. And you cannot make RBCs without making hemoglobin. Okay, so both synthesis of both will increase. Now capillaries should increase. Wouldn't it be a good idea also to increase angiogenesis? Angiogenesis means making new blood vessels, new capillaries. Now you're not going to make new aorta, of course. Okay. When you talk about angiogenesis, it simply means making new capillaries. Making new capillaries. Let me give you an example. When you run daily, the demand for oxygen increases in your myocardium, in your heart muscle, because it's working hard. So to compensate for that, to increase the supply, the heart blood vessels in numbers, they will increase. You'll have collateral circulation. You'll have increased blood vessels, not arterial, not arteries, but capillaries, the smaller blood vessels. Sometimes arterial as well, but capillaries happens first. And this is why people who work out a lot, not necessarily lifting, but cardio, those who do more cardio, even they can have an artery that's blocked. They'll be asymptomatic because they have more blood vessels, more roads, newly formed roads or tubes or airways or whatever that can transport blood supply, for example. So they are prone to heart attack in that sense. They will compensate collateral circulation. Yeah.
Erythropoietin, it's a sign for hypoxia.
Yeah. Yeah.
It's elevated,
Yeah.
so it's a sign for hypoxia.
The number one cause of secondary erythropoiesis is hypoxia. Primary means there's a tumor or something. Secondary means something else increased erythropoietin production or RBCs, and that's hypoxia number one by far. Yeah.
I know this happens later, but when does hemoglobin start incorporating the fetal part?
Now, if your adult hemoglobin, what's the adult hemoglobin? Let me finish this, I'll come back to your question, okay? So do you understand that bronchodilation should increase, RBC synthesis should increase, and capillary or angiogenesis should increase? For example, if somebody goes to a mountain called K2, it goes to the top very suddenly, or it goes in for example, in space shuttle or whatever, it goes suddenly. And you know what? Unless you have a mask or something, there's less oxygen as you go higher up. The reason there's more oxygen close to Earth, because oxygen has a weight. It has a mass, so gravity pulls on it. So the most oxygen you will find is where? Closer to the surface of Earth, at sea level. higher up you go, less oxygen, because the gravity decreases as you go away from the core of Earth. So when you go higher up, there's less oxygen. Now in this case, a person, when he goes higher up in altitude, what happens? He will experience hypoxia. He will experience hypoxia. Now to compensate, all of these would happen. Some are acute, some are chronic. In some cases, there can be sudden, like dramatic increase in vascularization or angiogenesis. But guess what? If that happens in retina, what would it lead to? Blindness. Yeah. So there has been cases, you know, unless you go gradually, you deal with hypoxia. So if you have neurovascularization in the retina, it can clog up the rods and cones, and you won't be able to see. Okay. Deep sea diving?
Yeah.
No. Deep sea diving is with nitrogen, nothing with oxygen. That's a nitrogen pressure. With deep sea diving, you are at risk of nitrogen emboli. Nitrogen embolus. Okay, so when you go deep sea diving, suddenly, unless you're trained, you can have nitrogen bubbles in your blood vessels, and that can clog up your pulmonary vessel. You can have pulmonary embolism. Yeah. Okay, so this is hypoxia. So understand in this context, this is how you should know hypoxia. There's many things. So, for example, if the problem is with this, it's called anemia. If the problem is with this, it is respiratory problem or acute respiratory failure or whatever, or asthma. Let's just say asthma, because asthma is what? Bronchospasm, bronchoconstriction. Very good. Your bronchi, especially your airways, bronchioles. Asthma is disease of airways, especially which one? Bronchioles, arterioles. Bronchioles. They're sensitive to little allergen and stuff like that. They constrict. So that's for that. Diaphragm. What do you think? Maybe medication like cholinergic blockers. Medication that blocks receptor on diaphragm, or trauma. Seizures, yeah, absolutely. Seizure will do it too. And then alveoli, COPD, emphysema. A common cause. Emphysema is destruction of alveoli. Destruction of alveoli. And then heart, okay, all the heart diseases. Okay, so just to kind of give you. Yeah.
Will that be an increase in pH or decrease in pH? Will that be alkalosis or acidosis?
Excellent. Now it depends. Okay. If the problem is anything other than the lung itself, such that the lung, if the respiratory rate is increased, okay, if respiratory rate is increased, it will lead to alkalosis. If respiratory rate is decreased, it will lead to acidosis. So it depends what's the cause. Okay, of hypoxia in this case. Yep. Yeah. Okay, I know what you're talking about.
Versus ventilation.
Yeah. You see, I'm glad you asked that question, because now it relates to something. Ammonia combined with carbon dioxide. What do you get? Carbamoyl phosphate, right? Carbamoyl phosphate. The source of carbon dioxide is what?
Bicarbonate.
Bicarbonate. So you can say the same thing. I can use carbon dioxide here or bicarbonate. It's the same thing. Bicarbonate is a huge buffer. But carbon dioxide plus water gives you bicarbonate plus hydrogen. Whoa, not water. Okay, so I just wanted to do that. Now I can erase this part. Okay, so just know that carbon dioxide, you can substitute it by carbonate. It's the same thing. Okay, so in urea, the carbon could come from the bicarbonate or carbon dioxide. It's the same thing, because they go back and forth. Carbon dioxide in blood goes back and forth. Okay. Now, water plus carbon dioxide gives you proton and bicarbonate. Acidity means increase in protons or hydrogen ions, positive ions. This is acidity. Okay. Basicity means increase in hydroxide ion, which we don't have here. Okay. Now, when we have increase in carbon dioxide, that means the reaction will shift towards this way, right? Okay, when it goes that way, look, there should be a balance between this reaction. That means both sides should be equal. Let's put it this way. So there is a bucket, and this is connected by a membrane or a tube to this side. Okay, so hopefully this will make sense. This bucket is full of carbon dioxide and water, and this side is full of bicarbonate and protons. If I pour more carbon dioxide in here, this area fills up. What will happen to this? There will be a shift, right? Okay, so there will be a shift, and this will go here. So by increasing carbon dioxide, what are you increasing? Protons. Acidity. Protons in acidity. That's how it increases. Now, of course, if you breathe faster, what happens to carbon dioxide in your blood? Decreases, and the reaction will shift this way. So that will lead to what? Alkalosis. Okay, so that's what we're talking about. So it depends on breathing. Now, lungs clears carbon dioxide out of blood, and kidney will clear bicarbonate out of blood. Okay, so when you have kidney problems, it can lead to acidosis as well. When you have lung problem, it can lead to acidosis as well. Okay, so it depends. Okay, anything else? Fetal hemoglobin. So we have adult hemoglobin, or hemoglobin adult, and then we have hemoglobin fetal. Okay, what is the genotype, if you want to, of adult hemoglobin? All hemoglobin should have 4 subunits. Very good. Do you remember when you talked about proteins?
Yeah, tertiary.
Very good. So if you have one polypeptide chain known as alpha, another one known as alpha, another one known as beta, another one known as beta, when they all combine and make one large protein, that's quaternary structure. Remember that? Okay. We talked about primary protein structure, secondary protein structure, and tertiary. Tertiary is the polypeptide that is folded on itself. So there's alpha polypeptide, okay, and there's a beta polypeptide, and then there's delta or gamma polypeptides. So hemoglobin is made of, number 1, it's made of heme plus globin. This is hemoglobin molecule. Again, we're going it's not on the exam, so I'll just go over it quickly, and then we'll start with our own thing. We'll discuss it in RBCs anyway. Heme is a ring. Globin is a protein. Heme is a ring that should look like this. It's a lipid soluble ring. In the center of it is iron, and this iron carries oxygen. So who carries oxygen? Is the heme group, or the iron of the heme group. Iron is plus 2, plus here. Oxygen is negative 2, negative here, so it binds it much better. Okay, so that's why iron is the central molecule. You heard of iron deficiency anemia, correct? Yeah. Okay, very common in female, because they lose blood, and when they lose blood, you have no storage of iron, so you lose a lot of iron. Iron cannot be stored in the body, so you always depend on what? Dietary iron. Once you lose it, that means you have to replenish it much faster. If you can't, you will have iron deficiency anemia.
It can't be stored somewhere that you can't retrieve it.
There's no storage in blood, so there's no stores available. You can, not you. In theory, practically no. Yeah, that's why there's iron deficiency anemia. Yeah. So now the heme part we're not concerned with, because he wants to know the difference between globins. Globin comes in different chains. Okay, the globin molecule, there's one version known as alpha globin, there's another version known as beta globin, and there's another version known as gamma globin, and there's a fourth one, delta. So the globin chain comes in different shapes. Maybe the sequence of this chain is A, just for the sake of argument. Maybe the sequence of this one is A, and then suddenly T, C, G. Maybe the sequence of this is gamma is just G for gamma. I don't know. Okay? We're talking about amino acids, right? This would be glycine amino acid. This would be alanine amino acid. Okay, so we're talking about the sequence of this each polypeptide. So they're different. They're still globin chains, but yet they're different because they have different sequence. Therefore, they have different properties. Correct? Okay. Now, adult hemoglobin is alpha 2, beta 2. That's adult hemoglobin. Okay. Fetal hemoglobin is alpha 2, gamma 2. Okay, so there's a difference. Now the difference is not in alpha, the difference is what? Beta and gamma. Gamma has a higher affinity, a much higher affinity for oxygen. It loves to store oxygen. Okay, it loves to store oxygen. Beta has a lower affinity for oxygen. Affinity means how tighter you hold on to something. Loving. If you love something, you don't want to let it go. You want to keep it, right? For example, we love money. You want to keep it, right? Affinity in biochemical terms or organic chemistry, whatever term you want to use, it means keeping something, holding on to something, loving something. Beta has less love for oxygen. This is the people who want a charity and they want to give up, not Donald Trump. Right? Gamma is Donald Trump. He wants to keep everything. He wants to become president so he can keep even more. Just kidding. But who knows? Now, there's a huge difference. When you have these two hemoglobin, who is likely to hold on to oxygen much stronger? Fetal hemoglobin. And there is a membrane between the fetus, so this is the baby, fetus, and this is mom. They're separated by something known as placenta. And the hemoglobin is locked in within what? RBC. The hemoglobin is locked in within RBC. They're not just freely floating. Remember that. So the hemoglobins are within RBC. Oh God, help me. What happened to the hemoglobin? They disappeared. Let me quickly use a different color. So the hemoglobin are locked in within RBCs. If you understand this, then I think your question will be answered without even me answering it. Okay? Alpha 2, beta 2. Fetal was alpha 2, gamma 2. Now, when RBCs reaches to the area where there's less oxygen, it will give off oxygen. Okay? Now, but gamma hemoglobin has more affinity. So it technically extracts oxygen from who? Maternal RBC and holds onto it itself. This is why all this oxygen will diffuse from mom to fetal RBCs. Now, your question was, under what circumstances do we have gamma in adults? In adults who have sickle cell anemia, or it would work, that's one thing. Or let's say you have beta thalassemia. If you have beta thalassemia, beta chain is deficient, then gamma will come and replace it. But again, it's not such good in adults because what happens to this hemoglobin in adults? It won't let go of oxygen that easier. It holds on to oxygen. So even it goes to your tissue, it says, I can't give it to you. I know I brought the oxygen for you, but I'm not going to give you all of it. I'll give you part of it. So still your tissue can starve for it. But nonetheless, it works. So, yeah. So it's an alternative. We have genes. So of course on chromosome, just to help you understand more, whenever we think of these hemoglobins or the globin chain, they should be located on chromosomes. They should be located on chromosome because we never run out of it, right? If they're protein, that means the protein should have a gene. On chromosome 11, we have a gene for alpha chain. On chromosome 16, we have a gene for beta. Okay, so beta is located on chromosome 16. And 11 is located on, alpha on chromosome 11. Okay. So we always have this source. Let's say this gene is defective, and then I don't know where gamma is located. I think probably here. So we can produce gamma just to replace it. Okay, just to replace that, to compensate for that. Okay. All right, let's take a break, and then we'll